Greetings. Let's talk about how our cells make proteins. Proteins, remember, are one of the four different macromolecules that our cells use in order to build themselves and do the work required to be alive. Now, the nucleus of the cell is where the DNA is stored. And DNA contains genes, which are like the recipes for making all of the cell's proteins. So we can think of DNA as being like the cell's cookbook. The ribosomes are the chefs. They read the recipes and make the proteins. However, ribosomes are not in the nucleus. They are located on the rough endoplasmic reticulum or out in the cytoplasm. Also, the cookbook, the DNA, is too big to leave the nucleus on purpose because we don't want the DNA out in the cytoplasm mingling with enzymes that might break it down. So, the way to solve this problem is that the cell makes a copy of the recipe into what's called mRNA or messenger RNA for the protein that it needs to make right then. Much in the same way that if you had a friend over who liked what you made for dinner and they asked for the recipe, you wouldn't give them the whole cookbook, you would copy it down on a piece of paper for them to take with them. So, the recipe from the cookbook gets copied onto a smaller piece of nucleic acid, mRNA, and this is called transcription. Transcription really means just to rewrite in the same language. To transcribe is to write over. So we're writing the same thing, using the same recipe, using nucleotides to copy it down. So it's kind of the same language, even though it's RNA instead of DNA. Now, the recipe can leave the nucleus and it connects with a ribosome either on the rough ER or in the cytoplasm. The ribosome then follows the recipe and makes the dish, the protein. And this is called translation. In translation, we're changing languages from a nucleic acid language to a protein language, or nucleotides to amino acids. Okay, so to understand this, we have to do a little bit of breaking down the processes. Now, starting with DNA, which we looked at in the last topic, where we explored DNA replication, where DNA makes a copy of itself, or rather, the polymerase makes a copy of the DNA. This time, we're talking about not copying the entire molecule of DNA for cell division, but instead one gene, one section of the DNA that codes for a protein. Now, when that gene is copied from the DNA into RNA, messenger RNA in particular, this is called transcription. So we're going from nucleotide to nucleotide, same language. When the messenger RNA is converted into a protein, this is called translation. Here we're going from a nucleotide language to an amino acid language. This entire process is called protein synthesis, starting from the DNA, going to the RNA, to the protein. Now, let's look at some of the differences between DNA and RNA. DNA is double-stranded. Here you can see the light gray strand and the dark gray strand are intertwined or twisted together. RNA, on the other hand, is single-stranded. Both of them use a phosphate sugar backbone. So the nucleotides are made in the same way, a phosphate, a sugar, and a nitrogenous base. The sugar in DNA is called deoxyribose. Deoxy means that oxygen has been removed. RNA is made from ribose. And if you look, you can see how ribose has this OH group or hydroxyl group attached to one of the carbons, but in DNA, that oxygen is removed. There's actually a hydrogen here. We just don't draw hydrogen unless it's bonded to something else. So deoxyribose is literally ribose with an oxygen missing. Turns out this is very important because there are enzymes in the cell that can recognize the ribose and chop up a nucleic acid. So, by DNA removing the oxygen from this carbon, the enzymes can no longer recognize it, and therefore the DNA is protected from being digested. Okay. So, DNA gets its name from the sugar. Deoxyribo is the sugar, nucleic acid is the polymer. RNA gets its name from the sugar. Ribose, nucleic acid, polymer. Now, in both DNA and RNA, we use the nucleotides, or rather the nitrogenous bases, guanine and cytosine, and they form hydrogen bonds to one another because they are complementary. However, in DNA, the adenosine or adenine nitrogenous base forms it forms a complementary pairing with thymine, but in RNA, it forms hydrogen bonds with uracil. So you can see that uracil and thymine are very similar. There's really just a methyl group here that is missing in uracil. So it kind of makes sense that adenine would like them both. But this one is the only one that is connected to ribose, and this is the only one connected to deoxyribose. Now, transcription is fairly simple. If you understood DNA replication, transcription is basically the same thing, except that the enzyme here is called RNA polymerase because it is making a polymer of RNA instead of DNA. It uses RNA nucleotides, the ones made with ribose, and it only copies a gene from one strand instead of copying both strands and the entire molecule. Now, in transcription, the DNA strands are separated once again by the enzyme helicase, and the RNA polymerase adds RNA nucleotides that are complementary to that one strand. So across from T, we would have A. Across from A, we would have U. And remember, because RNA does not have thymine, it uses uracil, we have to put a U wherever there would be a T. I will say this, though, it's very confusing, and we tend to add way more U's when we do it this way. So if this is difficult for you, just put a T here when you're doing transcription, and then remember to go back and erase all the T's and put U's. Across from C goes G, across from G goes C, and so on. So the RNA, mRNA, is copied from one strand of the DNA. Then the mRNA is released, and the DNA strands reconnect. All right, and we are basically done with transcription, except that the mRNA has to be processed before it is translated. We call this processing post-transcriptional modification. So after transcription, it has to be modified. So we're going to take this strand of DNA that we just made, and we're going to separate it into regions. And to make it a little bit easier, instead of writing all of these letters over and over again, I'm just going to take the regions and color code them and mark the entire region with another letter. These regions, some of them are coding and some of them are non-coding. The regions that are in yellow are called introns. The ones that are blue are called exons. Exons are the parts of the mRNA strands that will be expressed or turned into protein. These are the ones that are coding. Introns are in between the exons, or in between the coding regions. These get recycled. As far as we know, they do not have a purpose, but that might change someday as scientists continue to explore. So we have these enzymes called splicing enzymes that cut the messenger RNA in order to separate the introns and the exons. The way I'm showing this in the cartoon is not accurate, but it gives you a pretty good feel for how it works. So different cells can put the exons together in different ways. One cell might use U, V, X, and Z and have that turned into a protein. Another cell might use VWYZ and turn that into a protein. And yet another cell might use UWXY and turn that into a protein. And what that means is that each variation of the messenger RNA exons that we put together can code for a different but related protein. So we might have an enzyme that does the same job in, say, neurons and liver cells, but it's modified slightly so that it can suit the purpose of that particular type of cell. All right, let's move on to translation now that we've modified our messenger RNA. And translation requires 3 types of RNA. The first, which we've already looked at, is messenger RNA. It's called messenger because the message from the gene goes from the nucleus to the ribosome. So it's actually carrying information from one place to another. The next kind of RNA is called tRNA. The T stands for transfer because this RNA is going to connect with a sequence on the messenger RNA and bring with it an amino acid. And so it's transferring the amino acid to the ribosome where it can be put together. I often think it should actually be called the translating RNA because this is the piece of RNA that is making the translation between nucleotides and amino acids. The last kind is called rRNA or ribosomal RNA. The ribosome is an enzyme that's going to do translation, but it consists of both a protein component and an RNA component. The RNA component is necessary to be able to communicate with the other types of RNA, and the protein component is required in order to catalyze the reaction that puts the amino acids together. So we don't really see ribosomal RNA in action. We just know that it's there in the ribosome. All right, let's look at how this works. So we know that the messenger RNA is what actually codes for the proteins. But we have 4 nucleotides, A, U, C, and G, and yet we have 20 amino acids that we use in our body to make proteins. So how does this code work? One nucleotide can't tell us or give us 1 amino acid because then we could only have 4. Right? So if A gave us an amino acid and U and C and G, 1 nucleotide, 1 amino acid, then we could only have 4 amino acids. All right, so what if we have AA and AU and AC and so on? Like 2 nucleotides gives us an amino acid. Well, that only gives us 16 combinations. So we're still 4 short of having enough for 20 amino acids. So if we look at 3 nucleotides and all those different combinations to see if that gives us 20, that actually gives us 64 amino acids. So that is enough codes to cover all of the amino acids. Each one of these sequences of 3 nucleotides codes for a single amino acid, and we call it a codon. Anytime you see ON at the end of a word in science, that means unit. So codon means coding unit. This is the unit of nucleotides that's required to tell the cell which amino acid we need. So we have 20 amino acids, but we have now 64 codons. So what that means is that some codons are going to code for the same amino acid. For instance, CUU, CUC, CUA, CUG, UUA, and UUG all code for the amino acid leucine. And we'll see that same kind of repetition in other codons. All right, so let's look at how translation works. So we've made our mRNA, it's been processed, and now it's found its way to a ribosome. And translation starts when the ribosome assembles around the mRNA strand. The ribosome is always made of two subunits, and they sort of sandwich around the messenger RNA. So here are the two subunits of the ribosome. They sandwich around the messenger RNA, and they always start at what's called the start codon, which is AUG. So here's our AUG. That is the signal to the ribosome that this is the place to park to begin making the protein. Now, what happens next is that the first tRNA arrives and sits in the first codon site. Remember, a codon is 3 nucleotides. So the ribosome is actually covering 2 codons right now. So here's our tRNA. And the tRNA carries a specific amino acid, in this case, methionine. And at the other end, it has a sequence of 3 nucleotides called the anticodon, because those are complementary to a particular codon. So this just means that they match, but this is not an actual coding unit. We can't read a tRNA and know anything other than what single amino acid it's carrying. But messenger RNA gives us a series of codons so that we know the entire sequence of a protein. So this is just translating it for us. It's saying that if you have AUG, which matches this anticodon, then you will get this amino acid. Right? And here's our start codon, AUG. So first tRNA binds to the first codon site. Then a second tRNA with a different anticodon binds to the second codon site. Now the ribosome is able to catalyze the formation of a peptide bond, which is dehydration synthesis between 2 amino acids. Right? And now the first tRNA releases its amino acid and leaves. Now, off of this screen, what's happening to that tRNA is that it's interacting with an enzyme that uses ATP, which is the form of energy that the cell is able to use, and it's getting connected to a new amino acid, a new methionine, so that it can be used again when methionine comes up in the sequence. Now the ribosome moves over by 1 codon. The next tRNA arrives and sits in, again, the second codon site. Right? The ribosome makes the bond between the 2 amino acids, and once again, the first tRNA leaves. What you'll see is that each tRNA is still only holding the amino acid that it can be connected to. So this tRNA cannot hold any of these other amino acids, only this one, valine. But the amino acid strand, or the protein strand, is growing all the same. Ribosome moves over 1. The next tRNA arrives. The ribosome makes a bond. The first tRNA leaves. The ribosome moves over. A new tRNA arrives. The ribosome catalyzes the bond. And the first tRNA leaves. And this continues over and over for a much longer strand than what I have on the slide here, with the amino acid strand or the protein strand continuously growing. When we reach the end of the strand, or the end of the coding region, right here, the tRNA that lands at this codon is known as a stop codon. So when this tRNA parks on that, it actually causes the ribosome to release the mRNA, protein, and tRNAs. So this messenger RNA, this gene, has been completely translated now. We have reached the end. So how do we know which amino acid goes with which codon? Now, the cell knows because it has tRNA, which are like the cell's code breakers. In other words, every codon has a tRNA with a matching anticodon that automatically carries the correct amino acid. But scientists had to work this out the hard way by adding amino acids and seeing which ones got used with which codes and so on. So when they worked it out, they put it together in either a table or a wheel. I prefer the wheel. And this is what's known as the genetic code, or at least this is the version of it that we can read. The cells don't need this at all. So here's how it works. For any codon, like AUG, we find the first letter in the innermost circle. So this begins with A, so we find the A. Staying in the same quadrant, right, that first letter is in, we now look at our second options in the next circle outwards. So the second letter is U, so we have AU. Now we're going to look only at the letters that are connected to that U. All right. And since the third letter is G, we're looking at this G, so AUG. And now we can read the name of the amino acid that's associated with that codon. We use the same thing for any other codon. Now, we always use mRNA's code, not the tRNA, not the DNA, because the mRNA, it has the coding units, the codons. All right, in real life, we don't wait for one ribosome to work its way across a piece of messenger RNA. In reality, one ribosome attaches and starts scooting along, and then another one attaches and is right behind it, and another one and another one. In this case, these are moving from left to right. This is the mRNA strand that you kind of makes this line you can make out. Each of these big chunky bits is the ribosome. And you can see the proteins, the growing protein strands coming off of the ribosomes. And you can even tell that they're getting longer as the ribosome reaches the end of the mRNA. This is called a polyribosome. The name isn't so much important, but it helps us to understand how we can make so many proteins so quickly. And that's by, not waiting for everybody to do it one at a time. All right. As you know already from talking about proteins when we learned about macromolecules, in order for a protein to be functional, it has to be properly folded. So we go from our primary sequence to a secondary structure where we have the alpha helices and the beta folds or beta sheets. Then whole regions move together based on whether the R groups are hydrophobic or hydrophilic. That's our tertiary structure. And in some cases, we have multiple proteins that bond together in what's called quaternary structure. And that's what allows them to have their final function. All of this happens for proteins that are going to be secreted to the outside of the cell. They're made on the ribosomes in the rough ER, and they travel in vesicles which fuse with the Golgi. The Golgi has proteins to help them fold and maybe attach some carbohydrate groups, and they move in vesicles from one to the next until they end up in a vesicle that goes to the plasma membrane, fuses with the plasma membrane, and releases the protein out to the outside of the cell. Proteins that stay in the cell to do the work of that cell are modified inside the cytoplasm. And all of this is done by proteins, other proteins that we haven't even named. There are proteins that help to package the vesicle and to get the vesicle to leave the endoplasmic reticulum. There are proteins that help the vesicle to attach to the Golgi. There are proteins that help to fold the proteins that have just been made and read the tags that say where it's supposed to go next. So proteins, in addition to being the focus of this topic, learning how they're made, proteins are also the things that are deciding what gets done in the cell and doing the work. Which I hope puts the idea of the nucleus being the control center of the cell to bed entirely. It's just a cookbook. The proteins make all the decisions and do all the work.